My name is Desterio Nyamongo, the Director, Genetic Resources Research Institute. It's one of the 17 semi-autonomous institutes of the Kenya Agriculture and Livestock Research Organization. Our mandate is to conserve agrobiodiversity, and that's essentially the work of the gene bank, where different ecotypes of crop varieties are conserved for purposes of use in research and posterity. The National Gene Bank of Kenya is located at Muguga, west of Nairobi, about 26 kilometers off Nairobi-Nakuru Road. The idea to have the gene bank established was mooted way back in the 70s, and this on the realization that we were losing most of our farmer land races, because with the introduction of hybrids and other high-yielding crops, farmers tended to abandon what they used to grow as land races, which you could call farmer varieties. So that were fast disappearing from the farming system. Again, because of encroachment into the natural habitats, we were losing a lot of plant diversity as due to changes in land use. So that realization, the Kenya government approached the then Federal Republic of Germany for technical and financial support, who kindly accepted to offer that support, and the establishment of the facility started in earnest 1983. First, rehabilitating the cold rooms that breeders were using in various commodity research centers, and then establishing the facility which we now have at Muguga. That facility became operational in 1988. The gene bank has the cold room, which is the conservation facility that is run at minus 20 degrees centigrade. There are two factors that enhance the longevity of seed. The low moisture content and the low temperature. So what we do at the gene bank is that we dry the seeds to very low moisture content. So we have a drying room that has the capacity to do that. We bring the moisture content to about 5% moisture level. And then after they have effectively dried, materials are sealed in hermetic containers that cannot allow seeds again to re-imbibe, and stored under minus 20 in a freezer. Breeders had their breeding materials, which they kept in what we could call short-term to medium-term storage facilities in their respective commodity centers. So the initial material came from those centers. Then after the facilities became operational, we embarked on collecting missions, collecting the land races from farmers, from farmer fields, and also collecting the wild crop relatives and other forages from the wild. Because we adopted materials which used to exist before our establishment, there are materials that are older than the gene bank itself, materials that were collected in the 70s, some in the 60s. Our major collections, especially in crops, are sorghums, millets, and that's because we are in the region that is a center of diversity for such materials, and those materials are the ones that are mostly grown in arid and semi-arid areas. Maize being also a staple food for Kenya, we have a huge collection of maize, but then the highest collection are mainly pastures, the forages. That's the forage legumes and the forage grasses. Sorghum germplasm is comprised of slightly over 6,000 samples or accessions, if you want to call them. Finger millet is about 2,000. Sesame is about 2,000. Millet is about 1,500. Forages together, they could account for over 30,000. The main users of the gene bank are the scientists, the researchers who get the materials to undertake their breeding programs. Farmers have lately become active users of the gene bank, and this has come around with climate change. Some of the improved varieties are out-competed, especially when you go to marginal areas, are out-competed by the traditional varieties. And on that realization, the gene bank has been actively involving farmers in undertaking what we call participatory evaluation and characterization of the materials. And the process, farmers tend to select the ones that they think have attributes that they prefer. These are the farmer preferred varieties. So the farmers have also become, lately, very active users of the gene bank. Anybody who is interested in the gene bank material can visit the gene bank. It's a public facility, and those materials are also freely available, save for the handling costs that we charge when you are accessing our materials. Otherwise, materials are given out free. We are open throughout, especially when it doesn't mean that when you visit, you have to access the cold room. We only access the cold rooms when we actually want to withdraw materials from there, or we are taking new materials for conservation. But in terms of visiting the facility, we are open throughout. As gene banks, we exchange materials. So what we undertake in what I've already referred to as participatory evaluation with farmers, when we are taking these materials out, either for characterization, regeneration, or seed bulking, what we do is we invite farmers to come and view the crop. Sometimes we ask them, we prepare a questionnaire where they will use set a criteria for which they will select those materials. Say for sorghum, what we did is we grew about 2,000 accessions, slightly over 2,000 accessions, and invited farmers on 3 different occasions to come and evaluate the materials by themselves, using their own criteria, what would they prefer. Farmers normally have their criteria such as the size of the head, which will point to yield, the earliness in maturing, which will then mean that particular variety will escape drought, or the shortness of the plant, the height of the plant, which farmers will prefer short to medium plants, medium high plants, because that then they make the harvesting quite easy. The seed color is another attribute that farmers prefer. Farmers, from their experiences, they have realized that brown or red seeded sorghum varieties are less attacked by birds, so they will go for those varieties. So, again, there are varieties that are more filling in terms of how much you eat and feel satisfied, and they take long to before they require the next meal. Such varieties are slow in energy release, so farmers will go for those varieties. And how they identify that one is through seed color. In situ means you are conserving where it naturally occurs. Okay, ex situ is when you take it from its natural environment and conserve it elsewhere. For crops or land races, for that matter, you will consider conservation on farm actually in situ conservation, because That's where crops naturally occur. So when we introduce varieties from the gene bank back to the farmlands, the natural landscapes, you are enhancing the in situ conservation of those land races. So in situ conservation is when we take them from their natural occurrence, like if it's a wild plant, then from where it occurs naturally to in situ conservation. There is also the element of conserving wild crop relatives in the farmers' fields. Again, that can be considered in situ conservation, but it's an area that, as a gene bank, but as the National Gene Bank of Kenya, we are yet to get it to because of the limited resources. Women are the custodians of genetic resources over the years, since them, because they are the ones who have nurtured that diversity, and they are our major partners in the conservation, and even the community seed banks that we're establishing, they are mainly involved. Okay, those was also a few men who are interested. The youth, we are promoting and encouraging them to also join, because they are the future of agriculture, especially in doing the encouraging to do agriculture, and the establishment of those community seed banks. Again, they have been our target in terms of enticing them to join and participate in matters conservation. Now, the Chile Bank provides the diversity. We are talking about climate change, okay? You may have a high-yielding variety that has been released and promoted by research, but because of the erratic nature of the weather, the erratic rains, drought coming in, floods, Gene Bank has the diversity required to mitigate those impacts of climate change, because at the Gene Bank you get very short, early maturing varieties, medium maturing varieties, and long maturing varieties. Now, when a farmer grows that kind of diversity, chances that they suffer a total crop failure are very low, because if the rain becomes inadequate, then the early maturing varieties will at least escape the crop. So enriching the farmlands, the agricultural landscapes with that diversity ensures that farmers don't suffer total crop diversity. And then within that diversity, there's also nutritional diversity, all right, which again contributes to food and nutrition diversity.